Hello and welcome to chapter 6.2. Okay, so to finish talking about DNA, we're going to talk about specific genes and mutations. So total DNA of human is 3 times 10 to the 9th base pairs. So only 3% of that actually codes for proteins. And remember, when people refer to DNA as the blueprint of life, really what they're talking about are the instructions to build proteins, which really make up so many parts of our body and help us to do many of the functions that we do. So really the remainder of the DNA, it used to be called junk DNA, but we know now that that's a silly phrase. It's really just uncertain what the rest of the DNA codes for or does. So one DNA molecule contains many genes, and one gene equals one polypeptide. Okay, so a change in the nucleotide sequence of a gene, and remember a nucleotide is that sugar, that phosphate group, and the nucleobase. Okay, those 3 components together make up a nucleotide. So a change in the nucleotide sequence of a gene results in an altered polypeptide, and that would be called a mutation. And most genes have several different variants called alleles, which originally arose by the process of mutation. And an example of a variant, so a gene that is a variant, which is called an allele, an example of that would be like in a pea plant, how there is a allele that codes for a white flower, and the same type of gene or allele that codes for a purple flower. And because those are variants, they're called alleles. So DNA controls all activities of cells because all cellular activity is controlled by enzymes, right? Proteins, which are coded by DNA. So DNA controls which proteins are made and in what amounts. And so in this way, like I was saying earlier, DNA is literally the building blocks of life for us. Protein molecules are made up of strings of amino acids. The structure and function of a specific protein is determined by the exact sequence of amino acids, as well as the structure of the DNA, or of the protein itself. And DNA controls a precise order of amino acids, so it is the instructions for building that protein, for which amino acids go in which order. And so this leads us to the triplet code, or the codon. So a sequence of nucleotide bases in DNA is a code for the sequence for the sequences of amino acids. And the code is a 3-letter or a triplet code, and it's called a codon. Okay, and so it's like, you can think of it as like words in the instructions. Okay, each 3-letter word codes for a specific amino acid. The code is always read in a single direction and only on one strand of the DNA molecule. So this leads us to protein synthesis. So the first stage of making a protein is called transcription. This occurs in the nucleus. A complementary copy of the DNA code from a gene is made by building a molecule of RNA called messenger RNA, and this is a cousin of DNA. And then complementary base pairs are used to make RNA. So here you see this table, there's showing the differences between DNA and mRNA. So in mRNA, there is no T or thymine. It is uracil instead. So A binds with T in DNA, and in mRNA, A binds with U. Okay, so T is replaced by U. Okay, transcription is followed by translation. And let me just go back real quick. I just want to say, so when you think about transcribing something, it's copying something, right? And that's what's happening here. In the nucleus, the DNA is being unzipped, and a copy, a complementary copy of one side of the DNA is being made into an mRNA strand, and that's why it's transcribing it, right? It's reading, it's transcribing, it's writing, it's creating and recreating the information on one side of that DNA. And then it's followed by translation. So translation is when the mRNA code is translated into amino acids. And this is done by tRNA, which is transfer RNA. So the mRNA leaves the nucleus and will find a ribosome in the cytoplasm. So tRNA molecules contain a triplet of bases, and this is called an anticodon, and it's attached to one of 20 specific amino acids. So the tRNA molecule will match their triplet, their anticodon, with the complementary mRNA triplet, which is a codon. And it's the same thing where one base pair is going to only bond with another base pair with its complementary base pair. So in this way, the tRNA molecules have an anticodon and they're carrying a specific amino acid. So when they go through that ribosome, they will find their correct codon and they will drop off that amino acid. That amino acid starts getting connected to another amino acid, which is connected to another amino acid, and then you have a newly born protein. I really encourage you to pause the video at this point and really study this, this model here. So when tRNA links with the mRNA, the adjacent tRNA molecules form peptide bonds between their amino acids. And this process occurs repeatedly until a complete polypeptide is made. And I want you to remember this is completely random. So these tRNA molecules that are coming through that have these amino acids, they're not coming into the ribosome in any specific order. They're randomly coming in, and if their anticodon happens to match the codon in the mRNA, it will drop off its amino acid. And if it doesn't, then it won't. Another will come through. And again, this will repeat over and over until a complete polypeptide is made. And usually several ribosomes will work together on a single mRNA molecule, forming a polyribosome. And if you look at this model, you can see that. So the red line is a strand of mRNA, and each of those globule bits are ribosomes. And then the polypeptides are the proteins that are coming out, right? That are being built. And so you can see here the direction of transcription.
Innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
Okay, so now I encourage you to rewind this, or go back in this presentation and look at or listen to the process of transcription and translation again, and then watch the video a second time. I've watched this video 100 times, and every time that I watch it, I just love to watch it. It helps me to understand and really visualize the whole process, which I think is really important. So again, I encourage you to sort of go through this whole process of learning about transcription and translation a second time. Okay, so here is a copy of all of the 20 amino acids. And so you can see here that on the left is the codon that calls for the amino acid, which is on the right. And these are obviously abbreviations for the amino acids, but it's interesting if you look, you can see that 2 to 4 different codons will call for the same amino acid. And I really want you to, and we'll talk about this in class, but I really want you to think about why that would be. Why would more than one codon code for the same amino acid? Okay, here is a wheel to help you understand how these amino acids are, or excuse me, how these codons are calling for each of these amino acids. Okay, so we'll talk a minute about mutations. So there's a few different types of mutations that you're expected to know. The first one is substitution. So this is an exchange of one base for another. So an example would be switching an A to a G. And substitutions can cause a change in the codon, and that codon can call for the same amino acid or a different one. And really it depends on the place and the nucleotide that changes, whether or not a change in the protein will be induced. So if there is no change in the protein, that is called a silent mutation. So the change in a codon that does change the amino acid, and therefore the protein, really can cause some problems. An example of a current disease that is the result of a substitution mutation is sickle cell anemia. So a change in an amino acid coding codon to a single stop codon can cause an incomplete protein. And this can have serious effects, since an incomplete protein probably won't function well. Okay, another is insertion. Insertions are mutations in which extra base pairs are inserted into a new place in the DNA. And as you can see in this model, and as you can imagine, inserting a codon changes every codon from the point where it's added on. A deletion does the same thing. It's where a mutation, it's where a codon is lost. And it could be one codon or 2 codons, but either way, it changes the rest of the codons after it. Okay, another is a frame shift. So since protein coding DNA is divided into codons that are 3 bases long, insertions and deletions can alter a gene so that its message is no longer correctly parsed. So if you, earlier when I talked about codons being like words that are making a sentence, which are the instructions for building proteins, if you consider the sentence, "the fat cat sat," each word represents a codon, okay? And if we delete the first letter and pair the sentence in the same way, it no longer makes sense. So in frame shifts, a similar error occurs at the DNA level, causing the codons to be paired incorrectly, and for the instructions for building that protein to be dramatically affected. Okay, and that is it for 6.2. We will see you in class.